we're going to talk about membrane transporters. And you've seen these are actually somewhat conceptually similar to ion transporters. Ion transporters will reappear in this lecture a little bit here and there. But I think you'll see that membrane transporters will go through and discuss what they are and how they work and the different types. But you'll also see that these are quite important for pharmacology because they're involved in things like ADME, kind of determining how much drug gets to particular places. And so there's some really important pharmacology implications too. And there are also some cases where there are drugs that have been developed against membrane transporters that do some really useful therapeutic things, like altering how much acid you have in your stomach, water retention, things like that. So pretty important stuff. And so we'll go ahead and start out with just some basics on this, kind of an overview of the situation. So as you know, all cells have membranes, and what you can notice here is that the membrane is a phospholipid bilayer, and basically there's an exterior part that's exterior to the cell and an interior face that is facing the cytoplasm of the cell. And the lipid portion of this, which is all sequestered into the middle of the membrane, is very hydrophobic. But phospholipids are structured so that parts that face the outside of the cell and the cytosol, which are basically aqueous, these are hydrophilic. And so in order for things to move into and out of cells, they have to somehow transit the membrane. And to do so, they encounter this very hydrophobic region. So for instance, a very charged molecule that likes to be with water is going to have a hard time passing through this by itself, whereas other things can transport through very readily, depending on the chemistry of the compound or the molecule that's transporting. So membranes make the biochemistry within the cell very different from the extracellular space. And many compounds actually have to cross multiple membranes in order to reach their targets. There's the plasma membrane around the cell, but there are membranes around mitochondria, around the nucleus. There are a lot of vacuoles within the cell. They're also surrounded by membranes. So membrane transport is really important for transport across membranes, and it's important for drugs as well. So how do molecules cross membranes? Basically, there are just two real ways that this happens. Some molecules can cross membranes passively. They can just diffuse through the membrane. And then there are others, and so, you know, even passive transport, where there's actually no energy usage to get molecules across the membrane, this can actually occur in two different ways, either completely passively or in a facilitated way. So one thing that really kind of drives transport across membranes are concentration differences. So if you have a compound or substance that is in high concentration on one side of the membrane and low on the other side of the membrane, there's going to usually be a driving force that wants to move that substance from the high concentration to a low concentration. It will enter. Now that can happen just purely on concentration difference, but if it happens to be a charged molecule, there's even more of a driving force. And of course we saw that on Monday when we were talking about electrochemical gradients that are really important in neurons, for instance. But at any rate, some things can actually just passively diffuse through, but there are another form of passive transport is facilitated diffusion. And this is a case where some sort of a membrane protein exists embedded within the membrane that allows diffusion to go through. And so a great example of this is just a pore. So a charged molecule, ordinarily, let's say a hydrophilic molecule, would have a very hard time just doing passive diffusion because to do so, it'd have to encounter the hydrophobic core of the phospholipid bilayer, right? But if you have a poor molecule that, and the pore is hydrophilic all the way through, then it's no problem for a hydrophilic molecule to just transit through that pore space and into the interior of the cell. Okay. So anyway, facilitated diffusion is kind of one of the main ways that a lot of larger molecules that might not be able to transit that hydrophobic core of a membrane, that's one of the main mechanisms for them to get through in a passive sort of way. But there are also cases where active transport is required. And so both active transport and facilitated transport require proteins, membrane proteins. And facilitated transport tends to also move molecules from a high concentration to a low concentration. But active transport mechanisms actually require energy, whereas facilitated diffusion does not. And so that's kind of summarized here. And these are basically the three major ways that molecules can transit the membrane, just to review. So diffusion is when the molecule is able to just cross the membrane without, it can just penetrate the membrane and get right through to the inside of the cell, into the cytoplasm. And that's a passive process. The other passive process is where a pore or a transport protein of some kind is involved, and you can just get facilitated diffusion, and it's the protein that does the facilitation, and that's also concentration dependent. But active transport requires energy, oftentimes in the form of ATP, and it is unique in that it can drive transport of compounds across the membrane independently of concentration. And so it can actually go backwards. These are both kind of downhill, going from high concentration to low concentration. But active transport can actually drive the transport of molecules against a concentration gradient. And of course, that takes energy to do something like that because it's not energetically favorable. And so ATP is usually the source of that energy. Okay. So there are a lot of transporters actually in most organs, but there are a few organs that are especially important for their transport capacities. And the thing that ties all of these organs together is the fact that there's blood that gets into all these organs. And so these things are basically adding things to the blood and removing things to the blood. And of course, that's going to be very important for drugs as well, because most drugs have to make it to the bloodstream in order to be active. But these are kind of some of the major sites of really important transport in organs. And we have here the small intestine, the liver, the kidney, and the brain. So the small intestine, of course, is where most drugs are absorbed. It's also where nutrients are absorbed. And so the cell type that lines the lumen, the inside of the small intestine, are intestinal enterocytes. And so these are epithelial layers of cells, and they can move compounds from the inside of the intestine through the enterocyte and then deposit them into the blood, okay? And it can also move, and you'll notice that there are transport mechanisms to take things from the intestine and then put it into the blood, but there are also ways that these cells can actually take compounds that are in these enterocytes and put them back into the small intestine, okay? So transport is bidirectional, both between these enterocytes and the inside of the small intestine, and it's also bidirectional between the inside of the enterocytes and the blood. And these are very polarized cells with a basal and apical direction. One side of these cells faces the lumen of the intestine, the other side interfaces with the blood. And so these are polarized cells, they're asymmetrical. And the two types of transporters are called ABC and SLC, and we'll talk about what those are in detail in a moment. So that's what's going on in the small intestine. There's also a lot of transport in the liver, but this is actually called hepatobiliary transport. And so if you think about it, a hepatocyte is the most important, or one of the most important cell types in the liver. And hepatocytes make contact with the blood, but they also make contact on the other side with biliary cells. So kind of an interesting thing. One side of a hepatocyte contacts blood, the other side contacts the bile system. And of course, and so you can have things that could enter from the blood via SLCs, for instance, and then get moved into the bile, and it can be excreted from the body. Okay? Cholesterol is a great example of that. It can be taken up from the blood into the hepatocyte, and then cholesterol can be metabolized into bile acids and salts and put into the biliary region. So again, we have a polarized cell, polarized in that it has one side facing the blood, one side facing another fluid, in this case bile. And again, SLCs and ABCs are important. And you'll notice that, by the way, the ABC transporters are ATP dependent, whereas SLCs tend not to be ATP dependent. Moving on to another important organ, the kidney. Once again, one side of a kidney cell faces the blood, and the other, in the context of the nephron, which is kind of the functional histological unit in a kidney, it faces urine. So these kidney cells can take up materials from the blood and put them into urine in the kidney. They also have the capacity to take things out of the urine and put them back into the kidney cell, like uric acid, for instance, which we'll talk about gout later. That's an important contributor to gout. And then the final organ I want to mention is the brain. And here we're talking about the vasculature in the brain, the blood that goes into the brain. And one side of these vascular endothelial cells that line the brain faces the blood. That's just circulating through the whole body. And the other side is the interior of the brain. So these are cells that are arranged into a very tight configuration. And there are tight junctions between these cells that don't allow anything to go across this easily. And typically we call that the blood-brain barrier, the BBB. And some drugs can cross the blood-brain barrier and some can't. But the ones that can all make use of membrane transporters to do so, okay? And so that's a very rigorous kind of controlled way. It has to be a special molecule to be able to make it from the blood into the interior of the brain. It's probably one of the least leaky of any of these membrane type barriers. Okay, so you already get the idea that this is how nutrients and drugs can get from blood into other sites. The blood is what ties it all together. But depending on the organ, you're talking about, the lumen of the small intestine, the biliary flow, urine, and crossing the blood-brain barrier. So a little bit of kind of anatomy and physiology there. We can further divide transporters into uptake and efflux transporters. So here is a membrane in a cell. We have an extracellular side and the intracellular side, the cytoplasm here. And uptake transporters take materials from the extracellular space and move it inside the cell. These can actually be reuptake transporters are also important. They are also moving things from outside the cell into the cell. But this is, for instance, important in neuropharmacology. One of the most important reuptake transporters is, of course, the serotonin reuptake transporter. And of course, the synapse is outside the cell, right? And reuptake transporters are taking neurotransmitters and putting them back into neurons. So that's going to affect ADME, the concentration of neurotransmitters in the synapse in this case. Then there are other transporters that are just purely efflux transporters. And these do nothing except take substances out of cells and excreting them out into the extracellular space. Okay? Turns out that's very important for chemotherapy resistance, and we'll talk about that too. Okay, so that's the overview. let's get into this. Active transporters. We've already seen this one, ion pumps, sodium, especially like the sodium-potassium exchanger pump. You can think of it actually as a transporter, but you remember that that is the one that exchanges sodium and potassium, and that we talked about that actually on Monday. But basically, when it can exchange sodium and potassium, but it uses ATP, and typically these are going in opposite directions. Every time 3 sodium ions are excluded from the cell, 2 potassiums come in. So we've already kind of seen that, but technically it's a transporter and it's energy dependent. Now here's a new transporter that's very important for stomach acid, and it's called the hydrogen potassium pump. Typically you call this the proton pump because a hydrogen atom is a proton, and so drugs like Prilosec are proton pump inhibitors. But where are these pumps? They're in the lining of the stomach, okay? And more specifically, they're in a special type of cell that is in the lining of the stomach called a gastric parietal cell. And these gastric parietal cells are very interesting. What they do is they're energy dependent. They use there's an ATPase activity associated with this pump, but they're exchanging hydrogen and potassium ions. More specifically, they're pumping hydrogen ions and releasing it into the stomach, and then they're taking up potassium ions. So it's another exchanger pump. But and this is open to the stomach down here. So if you're loading up the stomach with hydrogen ions, what's going to happen to the pH? It's going to go down. It's going to become very acidic, right? So basically, this is what makes the stomach acidic, and it's very important for digestion. And it turns out that there's a whole bunch of regulation that goes on with gastric parietal cells. So for instance, you don't want your stomach secreting a bunch of acid at 3 a.m. That would be unpleasant. But you'd like your stomach to secrete a bunch of acid after you eat a meal. And so there's a lot of regulation of these, of the gastric parietal cells, so that this is happening at the right time in the right context. But it's another interesting pump. And this is a really important drug target actually as well. Because if somebody has too high a stomach acid, you're going to have all kinds of problems. It can cause all sorts of pain, difficulties with digestion. If you regurgitate it, it can erode your esophagus. And so there are drugs that actually inhibit the proton pump that we'll revisit later before the semester is finished here. So there are other interesting transporters that kind of work as pairs, and they actually use ion gradient differences to drive transport of another molecule. So here is galactoside permease, and it's termed a secondary active transporter. But the way it actually transports lactose, for instance, is a very interesting mechanism. What it does is this is actually kind of the galactoside permease is actually a transporter for lactose, and I think it might do galactose as well. So this is a sugar, right? But in order for this transporter to work, it needs some driving force, and it turns out that the driving force is a gradient in hydrogen ion concentrations. So when hydrogen ion concentrations are high outside the cell relative to the inside, that kind of facilitates the lactose import into the cell. Well, how do you generate a hydrogen ion differential? It turns out that a proton pump is doing that, similar to the one we just saw in gastric parietal cells. But there is a pump that actually uses energy to generate a hydrogen ion differential between the outside and the inside of the cell, and that's essential for the co-transport of lactose. And I think when you get down to the molecular level, I'm not sure exactly how this works, but I have a couple of ideas. It's kind of an interesting mechanism. You remember how solvation can, thinking way back, solvation, water molecules can actually interact with ligands and receptors, kind of to make the fit correct. And I think there's something similar going on here, where lactose isn't able to move through this unless it's complexed in the vicinity with hydrogen ions. So kind of an interesting thing there. But ultimately, the ATP is used to create the concentration gradient, which is then required for the transport activity of this transporter for lactose. So it's kind of like a cooperative pair of proteins, right? One makes the driving force and one does the transport. All right, so ABC binding cassette proteins. Let's look at these. We have ABC and SLC, so we'll go through both of them. So ABC transporters exist usually as a dimer. Of course, they're embedded in the membrane. This is the outside of the cell, sometimes called the periplasm, has transmembrane domains, and then a number of domains inside the cell, intracellular loops, and importantly, there's an ATP binding cassette here. And this is that's why they're called ABC transporters, because they have an ATP binding cassette, and that's where ATP can associate with this transporter. And when ATP is hydrolyzed, that gives the energy to drive things through the pore of the transporter. So there's some really important pharmacology considerations here. They're really important in drug resistance, believe it or not. And so ABC transporters, there are many ABC transporters, but three really important ones are called MDR1, MDR1/2, and BCRP. So MDR stands for multiple drug resistance. So MDR1 and MDR1/2 are two different drug resistant transporters, and BCRP stands for breast cancer resistance protein. So what these molecules do is they pump drugs back out of cells. So great, you have a drug that works, you get it inside the cell, and these ABC transporters, these MDRs, actually pump the drug right back out of the cell, so it can't work, unfortunately. Why? So they're kind of like our enemies in terms of pharmacology, but they actually exist for a very important reason, and that's the export of toxins that get into cells. That's why they evolved, to remove toxins that you might encounter through plants out of your cells. And a lot of times when you give a drug, the cell mistakes the drug for a toxin, and it wants to pump it right back out of the cell. So they have kind of a reasonable mechanism. They exist as dimers, and they have closed and open positions. And what happens is when it's open on the cytoplasmic side, the ligand, the drug, for instance, can bind, and then ATP hydrolysis drives a conformational change, closes the dimer, and then it opens the top, and the ligand can be secreted to the outside of the cell. Okay? And then it turns out that once ADP is released, then it flips back into this ready position. Okay? So it's basically ATP hydrolysis driving excretion of a compound, and then release of ADP and inorganic phosphate, and that resets it to the open position. Yeah. So then they pump drugs back out of cells. So there's a very famous one, which is called ABCB1. That's the name of the gene. But the protein itself has been called P-glycoprotein for a long time. So ABCB1 and P-glycoprotein are the same thing. And PGP, or ABCB1, is actually also the same as MDR1, multiple drug resistance 1. So this has been studied by scientists for a long time, and it actually has three different names: P-glycoprotein, ABCB1, and MDR1, but they're all the same thing. So what happens, though, is that this drug, or this pump, an efflux transporter, actually takes drugs that are inside the cell. It also can transport toxins that are not drugs, and it pumps it right back out of the cell. So it's kind of a defense mechanism for the cell. It's expressed in the intestinal epithelium, it's in hepatocytes, the kidney, even in the blood-brain barrier cells, and it's in all sorts of cells. And so these are all anti-cancer drugs here, and it shows that, yeah, this transporter, P-glycoprotein, can remove them from cells. So there's a connection here with nuclear receptors. So a lot of nuclear receptors, these are, remember, receptors and transcription factors, they actually can be activated by drugs. They bind drugs as a ligand, and that turns on their transcriptional activation. And so PXR, SXR is a pair of heterodimer of transcription of nuclear receptors. And so when the drug ligand associates with this heterodimer nuclear receptor, it acts as a transcription factor, and it transcribes all of its target genes. Well, one of the target genes is the MDR1 gene, and that makes P-glycoprotein. So the drug actually induces the expression of the gene that encodes the transporter, P-glycoprotein. It's one of the targets. Yeah. So these are usually, so PGP is actually a drug efflux transporter, and it impacts the ability of drugs to engage with their targets. But it's naturally kind of a toxin resistance mechanism that exists within cells. So this is really important in cancer drug resistance. It turns out that a lot of cancer cells come to express high levels of P-glycoprotein, and so when you give chemotherapy, you know, an anti-neoplastic, a cancer drug gets in, and P-glycoprotein pumps it right back out. So there's been a lot of interest in coming up with secondary drugs that actually inhibit P-glycoprotein, so you don't pump the chemotherapy agent right back out. And that's met with limited success, I would say, but it's kind of a good idea. You inhibit the transporter that's removing the chemotherapy agent, so the chemotherapy agent is retained and can do its action. But PGP, P-glycoprotein, is really important in the development of cancer-resistant cells. And so the idea, so there's been a lot of interest in finding PGP inhibitors so you can maintain the efficacy, the action of chemotherapeutic drugs. But they've been tough in clinical trials. Just incidentally, looking back at this gene, I'm going to just put this on the board real quick. How does chemotherapy resistance happen? It's also genetic. So you have a tumor, and you have billions of cells, you know, in a tumor, and you hit it with a chemotherapeutic agent, a drug, and it kills these cells. A lot of them die, but cancer cells also can mutagenize very quickly. And what if there's a couple of cells in here that have upregulated their MDR1 gene, the gene for P-glycoprotein? Well, you kill the tumor, but you don't kill these cells because there's just a few cells that happen to be overexpressing P-glycoprotein, and everybody's happy, the tumor shrinks, and then 2 years later, these start to divide and you make a new tumor, but this time they're all highly expressing PGP, and the drug won't work on that anymore. So that's a real problem with cancer relapse that is important to be addressed. And there are some ideas on that. But one of the reasons that happens is that in addition to the drug activating the MDR1 gene, anytime you add a chemotherapeutic agent to a cancer, you're actually doing artificial selection in evolutionary terms. Any cell that's undergone a mutation that makes it resistant to the drug, you kill all the ones that are sensitive to the drug, and the very few cells that happen to have a mutation where you've upregulated an efflux transporter, those now become, they make the new tumor, and then the whole tumor is resistant to the drug. So it is kind of like artificial selection. Yeah, and that's a problem. One of the problems too with, they're called multi-drug resistant because these transporters transport specific drugs out, but they actually they're kind of a generalist. They will transport all sorts of drugs out. So once a cancer is resistant to one chemotherapeutic agent, sometimes it's resistant to a whole bunch of them because this transporter will accept any drug and just move it back out of the cell. So there's a lot of interest in actually resensitizing resistant cancer cells. So you want to make them, you want to resensitize them to the action of chemotherapeutic agents. One approach is to inhibit PGP. So that's a big field in pharmacology, is resensitizing drug-resistant cancer cells. Let's move on to SLCs. And SLC stands for solute-like carrier transporters, but most people just call them SLCs. And you know, there are a bunch of these. There are 395 of them in the human genome. And they do all sorts of things. And they're basically the main type of transporters in addition to the ABCs that we just talked about. The SLCs, however, don't need ATP for their action. So they can either be facilitative transporters or secondary active transporters that use like an ion gradient like we talked about. But they're all sorts of places. They're in the cell membrane. They're involved in transport across mitochondrial membranes and in vesicles. So there are just a lot of these. And they can transport all kinds of things. Ions, inorganic ions, sugars, nucleotides, purines, amino acids, fatty acids, neurotransmitters, and of course drugs. And so these are really important. They're involved in transporting nucleotide analog drugs, for instance. They're involved in reuptake of neurotransmitters that we can develop antidepressants for. So a lot of important things. They're also, you don't have to memorize this list, but I do want you to know that there are many kind of rare genetic diseases that are out there, and these are just a list of several rare genetic disorders caused by mutations in SLC genes. So these are transporter-based rare genetic disorders, and there are a whole bunch of them. Okay, and they all have to do with altered transport across membranes. They're also great as drug targets, so there's been a lot of pharmacology development against SLC transporters. Glucose transporters are SLC type, and so there's been interest in getting drugs to act on those to treat diabetes. Neurotransmitter transport, especially reuptake of neurotransmitters, that's mediated by SLCs. So the serotonin reuptake transporter is an SLC type transporter. Bile acid transporters, important in liver health. Salt transporters, some salts can be transported by SLC proteins, so that's important in the development of diuretic drugs that eliminate water through the urine. Uric acid reabsorption from the urine, that's also mediated by an SLC, so that's important for gout, which is an overaccumulation of uric acid. And peptide transporters, they can also transport peptides, and so there are peptides that are used as drugs. So these things transport all sorts of things. And let's look at a few examples here. So here is an SLC transporter that's involved in glucose transport in the intestine, SGLT1, and GLUT2, and so these things are actually moving glucose from your intestine into your bloodstream. And so there has been interest in trying to alter glucose uptake from your diet as a diabetes treatment approach. This one over here, so SLC5A1 is a sodium and glucose co-transporter, so sodium gets involved to drive this. But again, this one is also interesting. But at any rate, there are all these SLC transporters that work in the intestine. There are others that function in the kidney, and those are also of interest for diabetes, because if you can alter glucose transport from the blood into the urine, that can also affect serum, blood glucose levels. So there's been some interest in developing drugs against these. Here are some of these drugs. And so these are FDA-approved therapeutic agents, and they're usually employed when somebody is having difficulty responding to more of a baseline anti-diabetic drug approach. Okay, and they can reduce plasma glucose levels, okay? And they can cause that glucose to be excreted into the urine, okay? Okay, so more SLC stuff. So the serotonin reuptake transporter, and we've actually talked about this quite a bit, it's a reuptake transporter, but it happens to be an SLC. So it's the main one is SLC6A4. That's sort of the famous one. That's a serotonin reuptake transporter. SLC6A2 is a norepinephrine transporter. I think there's another SLC for dopamine. But basically the idea is, if you look at the steady state levels of a neurotransmitter in a synapse, that is controlled partially by release of the neurotransmitter into the synapse, but it's also controlled by reuptake into the synapse, into the neurons themselves, okay? So the amount of neurotransmitter in the synapse is controlled by deposition of the neurotransmitter into the synapse, and also how much of it is retaken up into the cell. And yeah, so it's been a great target for pharmacological development, especially selective serotonin reuptake transporters, drugs. Okay. And basically, 5-HT is also another name for serotonin. And so this reuptake transporter, which is an SLC type transporter, SLC6, if you inhibit that, then more serotonin accumulates in the synapse, and that is a good treatment for depression. So there are a whole bunch of selective serotonin reuptake inhibitors, SSRIs, fluoxetine, paroxetine, citalopram, sertraline, and there are many others actually. And there are also some drugs that can inhibit more than one transporter. So these are drugs that inhibit both serotonin and norepinephrine, okay? And they all have different uses. So as I just said, they basically reduce the clearance of serotonin, the reuptake of serotonin from the synapse. And what happens when you have more serotonin in the synapse? Well, it binds to GPCRs in like the postsynaptic neuron, for instance, the neurotransmitter receptors. And so if you have more serotonin in the synapse, you'll have more binding to serotonin receptors in the postsynaptic neuron, and that can actually modulate how that neuron behaves. So it's a it's kind of the major area of pharmacological development to treat depression. So there have also been interest in SLC transporters in the liver, in the biliary system. So bile acids are actually derived from cholesterol. So the hepatocyte can turn cholesterol into bile acids, and then it gets secreted into the bile. Well, the way that happens is that once bile acids and bile salts are made, there are transporters that take the bile acids and salts and put them into basically into the bile ducts, okay? And so overaccumulation of bile acids is actually very damaging to the liver. It's called cholestasis when that happens. And so drugs have been developed that, and there's also a lot of, interestingly, there's a lot of reuptake of bile acids from the intestine back into the bloodstream. And so for people that have, you know, problems with high levels of bile acids and salts accumulating in the blood, this can be modulated pharmacologically. Okay, so ASBT is the same as SLC10A2. That's just the name of one of these SLC transporters. But basically, people have developed inhibitors that work on these transporters for bile acids. And here are some of these. Okay. So diuretic drugs are another example of drugs that modulate transporter activity. And so here we are basically interested in drugs that will work in the kidney and move water into the urine so you can eliminate water. And this is important for people that have edema, the accumulation of water, which can be a side effect of cardiovascular problems, for instance. A number of things can cause it. But if you think about movement of water across membranes, that's an osmotic thing, right? So it's moving water across membranes. And so osmosis, secretion and uptake of water, is very dependent upon salts, okay? So if you have, you know, high salt accumulation inside of cells, that's kind of hypotonic, and water is retained in cells. It can cause edema. And if you remove that salt concentration from within cells, then water can leave the cells. And so SLC12A is a sodium potassium chloride transporter, and it's involved in salt transport in the kidney. And so when you basically come up with drugs that affect this SLC transporter in the kidney, which is actually involved in transporting, you know, salt ions, you're actually going to affect the osmolarity of the cells, and water can be secreted. Okay. So these drugs basically alter transport of salt ions across the kidney and the nephron of the kidney. They're used for the treatment of edematous states. Edematous means edema. And edema can happen, water retention can happen due to heart failure, liver disease. Of course, kidney disease would also do that because the kidney is really important in putting water into the urine. And so mostly drugs have been developed that are diuretics that alter these membrane transporters. So we're just going through a few kind of clinical cases here. Next, another kidney-related thing is gout. So gout is an overaccumulation of urea, actually uric acid, and it can cause like arthritis-like features and inflammation. And it's basically, gout is caused by an overaccumulation of uric acid. And it happens in the joints and soft tissues. It happens in the toes and sometimes in the hands. And so SLC22A12, also called URAT1, it's actually the uric acid transporter in the kidney, okay? And so what this thing is actually doing is in the kidney, it's actually scavenging uric acid out of the urine and putting it back into the bloodstream, okay? So there are drugs that have been developed that can inhibit this reuptake of urea from the urine into the kidney and hence into the blood after that. And so it's really a reuptake inhibitor thing, kind of analogous to SSRIs, except that in this case, instead of a neurotransmitter, you have uric acid, urea, which of course urine is called urine because it has urea in it. Okay, and that's what the drug looks like, but it's a inhibitor of an SLC protein that's involved in urea transport. It's called URAT1. It's been discontinued because of business reasons. A lot of people, gout is not as much of a problem as it used to be. Here's another one that's in development, but gout is not a huge problem, but many people suffer from it, and it's definitely a transporter-mediated disorder. Okay, SLC15, we're going to start to shift into drug delivery really quickly in the last couple minutes, but this is a transporter that transports peptides into cells in the small intestine. So there are some peptide drugs. And so one of the interesting approaches here, since this is a transporter of peptides, you can actually link drugs to peptides that are transported by this peptide, and the drug goes along for a ride along with the peptide and gets into the body. So this is a great mechanism to get a drug that ordinarily wouldn't cross the cell membrane into cells because you've hooked it up to an amino acid, to a peptide or an amino acid even, that kind of drags the drug into the cell. But the reason it happens is because there is a peptide transporter. Last couple of minutes, this is kind of a short little section, but we can get through it very quickly, but it's kind of important. Drug transporters. So drug transporters exist in many types of cells, and these are going to affect ADME, absorption, distribution, metabolism, and excretion, especially things like absorption and distribution and excretion, because that's the transporters are what are doing absorption, distribution, and excretion, not so much metabolism. But I just want to mention that drug transporters are really important in ADME and also in pharmacokinetics because of the following. If you have decreased transporter expression in a clearance organ, an organ that's eliminating drugs from the body, if you have decreased transporter expression, well, the drug will accumulate to high levels in the plasma, right? So since these transporters are removing drugs from the blood, if you have poor expression of these transporters, drugs will accumulate to high concentrations. And that can cause increased concentration, and it depends on whether that's good or bad. It could lead to toxicity because you have unexpectedly high levels. It could also decrease concentrations on the other side of a membrane. But the whole take-home message here is that if you have altered drug transporter expression, it's going to affect the levels of drugs that accumulate in the blood. So, and it's important to kind of consider that this can work both ways. So if you have altered transporter expression, that can lead to increases of drugs. If it's increased transporter expression, it could increase the levels. If this is a transporter that is, for instance, moving stuff into the blood, if you increase it, you'll have more. If it's a transporter that's removing a drug from the blood, you increase that, then you'll have less, right? So it depends on how the transporter is configured across membranes and how it faces the blood. But the take-home message is that altered expression of membrane transporters can increase drug concentrations or decrease drug concentrations in the blood. And then you have to ask yourself, is that good or bad? If it's a drug that is prone to causing side effects, increasing drug concentration in the blood will lead to more side effects. If it's a transporter that's putting drugs into a cell where that drug works in the cell, then increasing concentration of drug in the cell might actually be good. So in the one case, it can cause toxicity. In the other case, it could be great. And so I do want you to just kind of think about that. And it's kind of, you know, logical, but it makes sense that transporters are really important in drug concentration because they're affecting absorption, sometimes excretion, and distribution is very, you know, distribution is basically controlled by the accumulation of drugs in one tissue versus another tissue. So very important considerations there. Yeah, so these are all kind of, I just ask you to look through these slides and kind of think about it. You can actually, the last thing I'll say, one other thing, and then we'll go, is that these drug transporters are also sometimes involved in drug-drug interactions, okay? So here's drug number 1. It's taken into a tissue by a transporter, okay? Drug number 2 might be a drug that inhibits a drug transporter. So this drug here that inhibits a transporter, maybe it inhibits the transporter that takes the first drug into cells, okay? And then you have a drug-drug interaction, and you can get high levels of the drug being accumulated in some cases, or low levels, depending on how it's configured. So drugs that affect transporters is especially important to be careful because those can cause drug-drug interactions very quickly or very easily. They're especially prone to drug-drug interactions to participate in them. And I think that's it. Thanks for, yeah, I let you out a couple minutes early last time. Here I kept you a couple minutes later, but that's the way it goes. All right, thanks everybody.